Imagine breaking free from daily injections for something like diabetes or obesity. Just a single treatment that works for months. Sounds pretty futuristic, right?
It really does.
Well, today we're taking a deep dive into some genuinely groundbreaking research that's actually bringing this vision, well, much closer to reality. It's fascinating stuff.
It absolutely is. For conditions like obesity, type 2 diabetes, these are really complex multifactorial conditions.
Right, not just one gene causing the problem.
Exactly. So applying genome editing has been a massive challenge. And then you've got these really effective drugs like exenatide, but they often mean daily injections. That's a huge burden for patients.
Yeah, definitely. Okay, so let's unpack this development. We're looking at a discovery from the University of Osaka, led by Professor Kichiro Suzuki. They've built what they're calling an in-body pharmaceutical factory using genome editing.
That's the concept, yes. An in-body factory right inside the liver. It's revolutionary because they're essentially reprogramming the body's own cells, liver cells specifically, to continuously produce a therapeutic substance.
Instead of just injecting it periodically.
Precisely. And their target was exenatide, well, an optimized version called CH4. It's known to suppress appetite, reduce weight, but usually you need those regular shots.
So how on earth do you get a liver cell to, well, become a drug factory? That sounds incredibly complex.
It is complex, but it's also very clever. Basically, they engineered the liver cells to efficiently make and secrete this CEKA4.
Okay.
This meant attaching specific genetic tags to the exenatide gene. Think of them as instructions. One tells the cell, hey, make this and send it out. That's the secretion signal peptide, or SP.
Got it, like a shipping label.
Sort of, yeah. And another tag acts like molecular scissors, ensuring the drug is properly activated once it's made. That's the furin cleavage sequence, FCS. They found a specific combination, NGF, FCS2, worked best.
And how did they get this blueprint into the liver cells permanently?
So they used their own genome editing tool. It's called the HITI method. It's very precise. They used it to integrate this new CXF4 gene directly into a really active gene that's already in the liver cells.
Which gene was that?
The albumin-producing gene. The liver makes tons of albumin all the time, right?
Ah, okay, so they're piggybacking on a gene that's already working hard.
Exactly. They're tapping into the liver's natural high-volume production line. It ensures constant, stable production of the exenatide. It's a really smart way to get continuous output.
Right. But how did they deliver this genetic package just to the liver? You wouldn't want it going everywhere.
No, definitely not. They use tiny capsules called lipid nanoparticles, or LNPs. Think of them like microscopic delivery trucks programmed to go straight to the liver.
Targeted delivery.
Targeted delivery at its finest, yeah. It minimizes effects elsewhere in the body.
Okay, so they built the factory blueprint. They figured out how to install it using HITI, and they used LNPs to deliver it just to the liver. The big question then, did it work in the mice? What were the results?
The results were quite remarkable, really impressive. After just a single injection of these LNPs.
Just one shot.
Just one. They could detect the exenatide being produced in the mice's blood for over 6 months.
Wow, 6 months.
Yeah, confirming that the liver was acting as this continuous factory. And health-wise, the obese mice that got the treatment, well, their appetite was suppressed. They avoided significant weight gain.
Okay.
And crucially, their abnormal blood glucose levels improved. Basically, their insulin sensitivity got much better. It was a real shift towards better metabolic health.
So more than just weight, a fundamental improvement. That sounds incredibly promising. But, permanently changing cells, safety must be a huge concern, right?
Absolutely critical. And they looked closely at that. The good news is they found no off-target effects, meaning the genome editing didn't mess with other genes unintentionally. That's a big deal for precision.
That is a big deal.
Yeah. And blood tests showed liver function stayed normal. Also, importantly, the exenatide made by the body didn't interfere with the mice's own natural hormone secretion like GLP-1.
So it worked effectively without causing obvious problems.
Exactly. The effects were actually comparable to healthy mice on a normal diet. It showed high efficacy and a strong safety profile, at least in these studies.
So connecting this to the bigger picture, what does this really mean?
Well, it fundamentally tackles that problem of frequent injections, which is a major hurdle for many biopharmaceuticals. But maybe even more importantly, it opens up new possibilities for treating those really complex multifactorial diseases, things we thought were just too hard for genome editing before.
Professor Suzuki himself called it a groundbreaking achievement, saying it could greatly reduce the medication burden on patients. That really sums it up.
It does. Now, obviously, there are challenges before this could be used in humans. More safety work, optimizing it for people, figuring out costs. But the core concept, this in-body biopharmaceutical manufacturing, it's potentially transformative, a whole new approach.
So, thinking about what this means for you, listening, look beyond just obesity and diabetes for a moment. Imagine this idea an internal factory being adapted for other complex conditions, maybe chronic inflammation,
Yeah.
maybe even certain cancers.
Continuous treatment from a single dose.
Exactly. How might that completely change our whole approach to managing long-term health? Making these life-changing therapies maybe as simple as one visit to the doctor, something to think about.